Welcome to Lecture 11 in DIY Transcriptomics. In today's lecture, we deal with the topic of functional enrichment analyses, and we'll spend some time just talking about what the heck it means to do an enrichment analysis, why we do it, and then we'll spend some time actually going through and doing different kinds of enrichment analyses within R using the data set that we've used for the class all along. So I know I've shown you this schematic many times throughout the course. It's because I like to chart our progress as we move through these lectures and scripts. And so we're really at the point today where we're sort of turning the corner and we're on the home stretch of this course, right? So we finished differential gene expression analysis and we clustered those genes to generate modules of coordinately expressed genes in the last lecture. And that really then translates or moves very smoothly then into taking those modules and doing functional enrichment analyses. So I've always marked these steps along the way in different sort of colors and only realized as I was looking at this slide recently that it's really quite analogous to getting your different levels of certification in karate, right? And so you've been working your way towards this black belt, and today is the black belt. So really, doing functional enrichment analyses often marks a real sort of end to the first pass through your data where you start to get actual insight into what the genes are doing that you pulled out of the differential gene expression analysis. So let's talk about the goals for today. We're going to carry out two different kinds of enrichment analyses. The first is gene ontology or GO enrichment analysis using the modules we identified in that previous script. Then we'll carry out a gene set enrichment analysis using our full data set. Hopefully by the time I finish these intro slides, you'll understand the difference between GO and GSEA. And then we have to understand, in order to effectively leverage the GSEA algorithm, you need to understand a bit about the molecular signatures database, or MSigDB, and how to access this even directly within R. And I think this is a good time to then take a step back from all this enrichment analysis stuff. At this point in the class, you've produced a lot of different graphics and visuals. And so it's a good time to talk about how those form the basis of sort of building blocks, if you will, for assembling a figure for a talk, for a publication, for a grant. And what are some of the guidelines I think of in terms of how to construct a compelling figure. And then, of course, we're going to open, start, finish the step 7 script today. All right, we're going to spend the entire time talking about functional enrichment analysis. This is also frequently called pathway analysis or signature analysis. Really all means the same thing. It's referring to the attempt to go from this, a collection of genes that clearly have patterns to them, to some understanding of what those genes might be doing, right? What do my genes do? That's the question we're trying to answer. Now, we've tried to improve our ability to do that, or improve our resolution, by clustering the genes so that they group into these coordinately regulated modules, because we're going to, again, assume that genes that follow the same pattern of expression across a set of samples are more likely to be functionally related. And so by clustering our genes into modules, we improve our ability to detect functionally enriched themes in these genes. So we have two modules to sort of deal with today, and that's kind of how we'll approach things. But I think it's always a useful point to, at this stage and really around this time in an analysis, to really think about why you set out to do an RNA-seq experiment in the first place. Because if you felt like after doing differential gene testing, you sort of felt like, so what do I do with this? There's a chance you may feel the same way after doing functional enrichment analysis. You're going to get not one theme sometimes associated with your differentially expressed genes or that comes out of your GSEA analysis. You may get many, many themes. And so it becomes an additional layer of information, and one that can often still leave researchers feeling a little bit like, so what next? And what I would tell you is that what next has a lot of possibilities. It's one of the exciting parts about doing omics experiments. One of the reasons some of us, like myself, get hooked on doing omics experiments is because there is a lot to explore. So one possibility is, depending on your system and your goals and your interests, you may put together a story about the transcriptome, right? About regulation of gene expression in your system under the conditions you evaluated. And that can be a very compelling paper. But you also may, and these aren't mutually exclusive, endpoints, by the way, you may decide that that you want to use these data to identify key targets or hits from your RNA-seq study that are good candidates moving forward for mechanistic studies. And that could be knocking genes down, right? So we have a list of genes here whose expression is increased. Those could be targets for siRNA knockdown. We have a set of genes here whose expression is decreased. Those could be candidates for overexpression, ectopic overexpression with cDNA expression vectors. We could search this list and prioritize it in lots of different ways to really distill value out of it. One of those ways is to take just the genes that are changed by the greatest order of magnitude and then take that list and ask, are there genes here for which there are reagents that I can study these genes? There's antibodies, there's siRNAs available, there's transgenic mice or knockout mice. That kind of sort of resource-related filtering can really help you whittle down a large list of genes to a very small list of genes that you could get started on mechanistic studies for right away. And again, that idea of prioritization of hits for mechanistic studies and publishing a descriptive, transcriptome paper are not mutually exclusive. In fact, it's probably more often the case that people will do both. They'll start off with the transcriptome story and then move into mechanism. Okay, so functional enrichment analysis, two main approaches, at least that we'll cover in this class. One is gene ontology or Go enrichment. There's a few things you should know about Go. It requires an arbitrary selection of genes. You have to set some cutoff or selection that returns to you a set of genes out of the larger universe of possible genes. And that's great because it means it doesn't require any data. Presumably the data is what led you to the selection of the genes. So once you have the selection of the genes, you don't need the data anymore. So you just need gene identifiers to carry out a Go enrichment. You just need gene symbols or Entrez gene IDs or something like that. It's species specific, so gene ontology terms are used to annotate genes in a way that's species specific. And the user, you, don't control that vocabulary. So, the genes in this part of the network here, in this background picture, are annotated with the term cellular metabolism, and these are annotated with apoptosis. We didn't label those genes with those functional terms. That ontology is really handled by ontologists because it's a pretty complex process that requires a lot of knowledge and training. And so ontologists really manage that process, and that's all done through the Gene Ontology Consortium. That's in contrast to another equally popular approach for enrichment analysis called gene set enrichment analysis, or GSEA, that in contrast to Go, you do not need to pre-select your genes. You're not selecting genes like you did with Go enrichment. And that actually was touted as one of the main advantages of GSEA over Go when the first GSEA paper was published, is that it just, you can arrive in a more unbiased position to do the analysis because you're not pre-selecting genes. As a result, then it requires both identifiers and data because you're not just picking the genes. The GSE algorithm is going to need all those identifiers and the data to do a ranking to begin carrying out that GSEA analysis. It's not species specific, and I'm going to say that with an asterisk because it's often not species specific the way most people do GSEA in the Broad Institute's GSEA software. It's assuming your data is human, and if it's not, you actually have to kind of coerce your gene symbols to human for it to work. But the way we'll run it in R, it actually is species specific, and I think that makes it just a better, more accurate analysis. And then lastly, and I think this is really important, with GSEA analysis, users can control the vocabulary. And what I mean by that is the way GSEA analysis is carried out, you use signatures, and those signatures are curated in a database that we're going to access. For example, all genes associated with NF-kappa B signaling. But there's nothing to stop you from making your own signature. You can group together genes in any way you want, as long as there's some reason to believe those genes belong grouped together. It could be they're located in the same region of the chromosome. It could be they share a function in your particular system. But once you have a signature, a group of genes that are related, you can carry out GSEA analysis. So there is that option for customization or user control. All right. I also would be remiss if I didn't say there's a ton of web tools for carrying out both Go and GSEA. And there's also commercial tools like Ingenuity's pathway analysis or IPA. I have comments about both. First of all, the web tools for Go and GSEA, I think they take me just as long to explain and walk people through as doing this in R. We used to in the course actually run both Go and GSEA using web tools. It was kind of a way for us to get outside of R. I find we can do it just as quickly in R, and you'll have more power to expand on what we do if we do it that way. The other thing I'll say is that when you do this online, you tend to get graphics that aren't really up to the standards you're probably used to with ggplot. And so I think we get, by doing it in R, we get better graphics. In terms of commercial tools, you know, I know a lot of people use IPA. I don't find as much value in it. What IPA folks will tell you is that you get better, ontologies, better annotations of gene functions, because the staff that develop this software they manually curate the literature and develop a proprietary database of expanded functional annotations, and therefore they can deliver to you more accurate, more sophisticated results. But I feel we can get really good results in R for free. So I don't use commercial tools. And the issue with commercial tools as well is sometimes it's hard to document your workflow. Okay, so I mentioned there's a lot of tools out there, and actually there hasn't been, it might be surprising, it hasn't been as much of an effort as you might think to kind of benchmark these different tools. Actually, it's quite overwhelming if you're just getting into functional enrichment analysis. Tons of different tools. Some are R-based, some are web-based. It's not really clear how they're different. And in fact, in some cases, they differ dramatically in the underlying statistical approaches or assumptions, which really, I think, highlights the need for these kinds of benchmarking studies. So this is one paper, it's on the lecture page for today, published this year, that attempts to do that and provides some guidance on which tools to choose. I'll just highlight that many of these tools use what's commonly referred to as an overrepresentation analysis or ORA approach. And a lot of times, though not always, overrepresentation analysis leverages statistically a hypergeometric or Fisher's exact test. So if you want to understand how enrichment analysis works and catch a wide swath of tools in one sort of statistical dive, you can learn about these statistical tests and you'll have a sense for how they work in many cases. So I've got a 5-minute sort of quickie StatQuest video that I linked to on the lecture page for today about Fisher's exact test. Note that 2 of these are exactly sort of web databases that I mentioned, David and Panther. Probably many of you have heard of David. It's got by far the most citations here, but surprisingly had not been updated for a very long time, and that's since been remedied. But yeah, so these are 2 of the web tools that I referred to in the previous slide, and we actually won't be touching on these today. We'll be using a couple R packages, one called gprofiler 2, which actually connects with gprofiler, which is a web tool, but we'll be using it from within R, and then cluster profiler, which is a pretty extensive R package that can carry out a lot of different kinds of enrichment analyses, including typical ORA, but also GSEA, and has access to a lot of different kinds of resources or gene sets. So you'll sort of scratch the surface with Cluster Profiler today, and you can continue to explore it on your own. All right, so 2 types of enrichment analyses we'll carry out today: gene ontology enrichment and gene set enrichment analysis. So what is gene ontology? Well, it's a structured vocabulary for describing gene function. It's got over 50,000 genes annotated with terms, and that's just for human. And over 100,000 term-term relationships. And what I mean by that is much of the effort of ontology is not just annotating genes with terms, but actually understanding the relationship between terms. And so this gene ontology actually forms something called a directed acyclic graph structure, or DAG. So if you're interested in more about that, I've linked on the lecture page to more detailed reading about gene ontology. Okay, so here you can see we have over 500,000 different annotations here for genes in the human genome. We also have lots of annotations for rat and mouse. Again, it's species specific. So if you're working with an organism that is that is not in this sort of top 4 or 5, in general, carrying out functional enrichment analysis can be really challenging because many of the genes in your organism are not functionally annotated. So you're really going to have to choose your path forward from differential gene expression based on, magnitude of expression or significance, availability of reagents, some of those other criteria that I mentioned. All right. So most of the gene ontology information that we'll be leveraging comes from the Gene Ontology Consortium, and I mentioned them before, and they involve a team of ontologists that manually curate to expand this ontology for a number of different organisms. All right. I think one key thing for you to understand is that Go is really 3 different ontologies. And those 3 different ontologies are Go CC, Go BP, and Go MF. CC being cellular component, BP biological process, MF molecular function. And these 3 ontologies really are meant to address 3 separate sort of questions or use cases. Go CC is really meant to tell us where a gene product is localized in the cell. Go BP then tells us what function that gene or gene product carries out in that location. And Go MF tells us how it carries out that function. In other words, via what molecular mechanisms. So I think a lot of times when people think of function, they think of BP. So a lot of times when people do Go enrichment, they actually focus on doing their enrichment analysis on just this BP collection. But there's times when you may care about cellular component or molecular function. If you're trying to ask, for example, if genes in your set of differentially expressed genes are enriched for transcription factors, you'd like to know if you have an enrichment of the term nucleus, right? So there's different ways to leverage these ontologies depending on your question, and that's why it's important that you understand there are 3 different ontologies. All right, so as an example, we can take a gene, eomesodermin, a gene involved in development, but also a critical gene for CD8 T cell development. And we can see that EOMES has an annotation for cellular component that is nucleus. Now, if we were talking about one of the STAT transcription factors or other transcription factors that reside in the cytoplasm until they're activated and then translocate to the nucleus, in those cases, there would often be 2 cellular component annotations, nucleus and cytoplasm. We can see that EOMES also has a number of BP annotations, including eoderm formation, mesoderm formation, brain development, and CD8 T cell positive differentiation. Remember, molecular function tells us what molecular events are involved in carrying out these biological processes. So we can see that is DNA binding, sequence-specific DNA binding, RNA Pol2 activating transcription factor binding, etc. And so maybe one thing you've noticed is that there's some terms here that sound a lot alike. DNA binding sounds a heck of a lot like sequence-specific DNA binding. And so that's one thing to keep in mind about all enrichment analyses is that Go is redundant. And as a result, it often requires some manual or automated way of trying to distill or summarize our enrichment results to reduce this redundancy. Otherwise, if we just took the top 10 Go terms annotated or enriched in our data, we might have 10 terms that share a whole lot in common and aren't really scratching the surface on our enrichment result. So finding ways to reduce that redundancy can be useful. So we won't really get into that today. We're going to work on just running these tools, but it is something just to be aware of. I mentioned David already as an online tool. You can absolutely check that out. We won't talk about it today. All right, so that's the crash course on gene ontology. What about gene set enrichment analysis? I told you you don't need to pre-select your genes, so you need to provide both genes and data for your whole data set. It's not species specific, and you get to control the vocabulary. A common web-based or software-based interface for carrying out GSEA was developed by the Broad Institute. It was originally the Broad Institute that published the first GSEA paper, the paper that described this algorithm and approach. It's also the Broad Institute that maintains the complementary database of signatures that you kind of turn to carry out the gene set enrichment analysis. So obviously they have web tools and software for doing that. The disadvantages of using the GSEA software is it's a lot of file formats to deal with. And it's kind of boring to talk about in a talk and to walk people through. But you should, rest assured these are just simple text files and they're reasonably easy to prepare in Excel. Again, we won't be using the GSEA software, but many people do. I feel like we can make just as good a graphics and do the analysis faster in R. So I told you Broad Institute also maintains a database of signatures. That's called the Molecular Signatures Database, or MSigDB. And there's a few key things to understand about MSigDB. I'm going to head over there now just to take a look. The first thing is that it's your one-stop sort of shopping place for signatures, that is, groups of genes put together because they share something in common. And they're curated. So somebody's put these lists together for you. And they've then taken the signatures or gene sets, and I'll use those terms interchangeably. They've taken the signatures and grouped the signatures into these sort of meta groups called collections. And so the MSigDB version 7.1, the one we'll work with, has about 7 collections, C1 through C7, plus this hallmark collection, which we'll kind of ignore for now. And the collections have some themes to them. They're not just random groupings of gene signatures. C1, for example, are genes grouped together based on their location on each chromosome. So you might find 50, 100, 300 genes in one signature that are there because they're all in the same location on chromosome X, for example. Collection 2 are curated gene sets, and they're pulled from a number of online databases, including Biocarta, KEGG, and other databases. C2, the collection number 2, is, I think, really the one most people think of when they think of functional enrichment analysis. It's pathways, all right? Collection 3 is based on gene target predictions for microRNAs and predicted transcription factor binding sites. So you could ask whether your differentially expressed genes are enriched for, whether your data is enriched for genes with an NF-kappa B binding site in their promoter. The collection 4 and collection 6 deal a lot with cancer-related gene expression data. So they're ones that I don't deal with as much because I don't work in cancer-related gene expression studies. Collection 5, if to really kind of blow your mind here, is all genes grouped together based on having the same gene ontology terms. So that's right, you can do Go enrichment using gene set enrichment analysis. And then collection 7 is probably one of the most recent ones, and it's a grouping of genes. It's a collection where genes are grouped together into signatures based on being immunologically related. And there's a paper I linked to on today's lecture page about how this collection was assembled and kind of how to interpret it. Okay, back to the slides. So we'll be pulling from MSigDB, and I'll show you a couple ways to do that once we get into the script. So how do we think about GSEA? So I'm going to try to explain this to you using a couple analogies, but let's first walk through what actually happens when you run GSEA. And this would be the same if you ran GSEA on the Broad Institute's sort of software, or if you ran it using other tools that run GSEA algorithms. So what happens is you choose 2 phenotypes. In our case, it would be healthy and disease. And the first thing that happens is GSEA will rank your rows, your genes or transcripts, in order of magnitude change between the 2 phenotype classes, between your 2 groups. So in our case, this would be genes turned up with disease, the highest gene at the top. Descending from there, the genes around the middle of your list would be genes that really aren't changed between your 2 phenotype classes, and then all the way down to the opposite, which is genes that are really turned down with disease. Once you have this ranking, the data is no longer necessary. Now we've rank ordered the genes, and that's what's used for the GSEA analysis. Once we have that ranked ordering of genes, we can take any single signature, let's say genes involved in NF-kappa B signaling, And we can start to march down our list of ranked genes and look for the NF-kappa B, the genes involved in NF-kappa B signaling, to see where they fall in my ranked list of genes. Now, the first thing that should imply to you is that our identifiers here should match the identifiers in the gene set collections, or else we'll never see any of the signature genes in our data because the identifiers won't match. So sometimes you get into these, you need to make sure your identifiers match when you're doing these analyses. But if we see NF-kappa B signaling related genes at the top of our list, then that tells us that those genes are sort of, involved in the disease process we're studying. If we see them at the bottom, maybe they're turned down by the disease process we're studying. And if they're just sort of randomly distributed throughout, then maybe they're not involved in our process that we're studying at all. And so as this process of looking for genes in a single signature proceeds, you assemble a running sum statistic. And so this common graphical representation of a GSEA result really has taken our ranked list of genes and tipped it onto its side. That's the colored bar here, with genes that were at the top over here, now on the left, and genes were at the bottom on the right. And anytime one of the genes from the signature, in this case this was evaluating a single signature, and it doesn't matter which one, a vertical line was struck anytime they saw a gene. So what you can see is whatever signature this was, there were a whole lot of genes found at the top of the list, suggesting a strong enrichment in our treated group or disease group, whatever you want to call it. How does that running sum statistic, how does that really work? One of the analogies I could give you is sort of almost like counting cards, right? So we can think about anytime we see a gene at the top of our ranked list, we add to that running sum statistic. If we see a gene at the bottom of our list, we take away from it. And so by sort of keeping that running tally, we can plot a curve here of an increasing running sum statistic where many genes were seen at the top. This is sort of analogous to counting cards, where somebody at a blackjack table who's really, hoping to get a face card may keep a running sum statistic in their head as they see the dealer deal cards to themselves and everybody else at the table, where anytime they see a low card, they add to their running sum statistic. Anytime they see a mid-level card, they don't change the running sum statistic at all. And anytime they see a face card, they subtract one from their running sum statistic. And so as their total running sum statistic gets high, the probability that the next card will be a face card is high, right? Because they've gotten a lot of, in order to have a high running sum statistic, they've gotten a lot of low cards and not many high cards. So the probability of a face card coming next is higher. So that's kind of the basic idea of count. Not that I'm a card counting, gambler or anything, but that's the basic idea. And the way, casinos deal with that is to use 5-deck shufflers and to change out dealers and change out cards. So not unlike how we think about accomplishing the running sum statistic for GSEA. We see a gene at the top of our list, we add to that running sum score, maybe see it throughout the middle, we don't change the score at all. Gene hits the bottom of our list, it takes away. So we end up with a high running sum statistic early on. It means we've got a lot of genes enriched at the top of our data set. Hopefully that gives you an idea of how this works. Now, that's for one signature. That process is then repeated for all the signatures we choose to evaluate. All right. Another really critical thing to understand about GSEA is it comes in two main flavors. The first flavor is self-contained gene set enrichment analysis. It's frankly one of the least common. We'll run it today and we'll compare it with the other flavor that I'll tell you about in just a minute. And I think you'll see why the differences between the two flavors are important to understand. So the self-contained gene set enrichment analysis really addresses the question of, are any of the genes in my set or signature differentially expressed? Are any of the genes in my signature differentially expressed? I don't think that's the question most people think they're asking when they run GSEA, but that's the question you're asking if the GSEA approach you use is called a self-contained approach. And it's called self-contained because it does not consider genes outside of your gene set. So if we're talking about genes involved in NF-kappa B signaling, it's just asking whether any of those genes in the NF-kappa B signaling pathway are differentially expressed in your data. That's in contrast to what's called competitive GSEA, which I think is what most people think of when they think of GSEA. And that addresses a different underlying hypothesis or question, and that's, are genes in my set, in my NF-kappa B signaling set, more commonly differentially expressed than genes outside the set? So that's why it's called competitive, because it does consider genes outside of your set. And that has real implications. If you have a ton of differentially expressed genes, then it could be hard to find any signature that meets these criteria, that is, being more commonly differentially expressed than genes outside the set, because you may have a lot of genes that are differentially expressed. Okay. So this is definitely the most popular, and I think whether people run this or this, they think they're running this. All right, so how do we know about significance then? How do we get a p-value with gene set enrichment analysis? And the answer is we frequently have to do something called permutation to determine if an enrichment score is actually statistically significant. One approach is to permute your samples. In other words, you take your sample labels, in our case, healthy 1, healthy 2, disease 1, disease 2, and you scramble them. You pretend you mislabeled the tubes. And then you redo that ranking that I showed you for the GSEA. You redo that, assuming those mislabeled samples are truth. If a signature was really enriched, truly biologically enriched in your data, then doing that sample scrambling and re-ranking, that signature probably should lose its enrichment, right? Because now it's, your disease and healthy are all mixed up. But as you can imagine, that's really not effective unless you have big groups where you could do these kinds of scramblings and really change things up. And so the idea of permuting samples, although preferred and appealing statistically, it requires a really large N, which most people don't meet. And that is that you need to have at least 7 per group. If any group has less than 7, then this idea of permuting sample labels isn't really tractable. The common alternative is to permute genes, and that is to scramble the genes and then rerun your enrichment analysis. So you still rank by your phenotype, by your samples. You're not scrambling your samples, but once you have the ranking, you would then scramble the rows, the genes, and the gene labels, and then run your enrichment, and again, marching down your list, asking whether the gene symbols are there. So you can see why this is a great way to kind of do that testing for significance, but it's subject to something called intergene correlations, which I won't get into here, but I've linked to these 2 papers, at least on the slides, so you could read more about that. And that can have in some data sets, a pretty substantial effect of inflating your type 1 error. That is, the chances of making a false positive, of having a gene signature enriched when it's actually not enriched in your data. Okay, so at that point, I think it's a good time to sort of go to our R script and start working on these methods.